Hello, welcome to the next lesson from the conscientious biologist, Ben Gallagher. This is the second one in the enzymes playlist. The first one covered the mechanism of enzyme function, which we've also done at GCSE, and it's not a massive step up on this area to study it at A-level. This one looks at factors affecting enzyme function, which again, you will have done at GCSE. This one just adds in the A-level details, the A-level terminology from what we've done on protein biochemistry and protein synthesis. There's also a little bit in this one on the effect of inhibitors, which didn't come up at GCSE. As always, please do subscribe to my YouTube channel if you haven't done so already. Do give this video a like if you enjoy it, and do follow me on Instagram and Facebook for lots of additional bits of biology to supplement your course. Thank you. So first up, the classic factor, temperature, comes up all the time in GCSE. You probably have to answer a question on it in GCSE, so you ought to know this already. So we're going to go quite quickly. So if we pull up a graph here with temperature along the bottom and enzyme activity along the side, the obvious start point is that we increase in the enzyme activity as temperature increases. Now, to explain that, I'm just going to put key words over there that you must include in your answers if you're going to get full marks. Because again, you've done it at GCSE, but lots of people go, oh, I know this. And then they write an answer that's correct, but so vague and so lacking in key terms that it doesn't actually score any marks. So make sure you do pay attention to the key terms. So over on that side of the graph, it's basically about collision theory. We're talking about the extra heat giving lots of extra energy to the particles. With that extra energy, there's going to be an increase in movement. If they're moving more, they're more likely to collide. And we're talking about all the collisions, so enzymes with enzymes, substrates with substrates, and enzymes with substrates. But even a collision between an enzyme and a substrate has to be kind of round the correct way so the active site and the substrate can engage. So not all enzyme substrate collisions will be successful. It has to be that collision between the active site and the substrate itself. But if you've got more movement and more collisions, then you're more likely to get that link between the active site and the substrate, more likely to form an enzyme substrate complex, which of course will give you more products, which is how you measure enzyme activity, by how fast they create their products. So that sort of side should be really straightforward. But then there's very much two halves to this graph. As the temperature increases more, and we'll move this into what we'll call section B, you can see a rapid decrease. Now that decrease is because the enzyme denatures. Now denatures, remember back to where we studied in protein biochemistry, the video lesson, how proteins fold. They go from their primary structure of the polypeptide through secondary and tertiary or quaternary, depending on the number of polypeptides and prosthetic groups. But that final shape gives them their function. If an enzyme is heated too much and denatured, what we're basically saying is that it melts. Specifically, we're saying that the polypeptide or chain of amino acids unravels because the hydrogen bonds, remember they were so important in the secondary structure, they're going to start to break, unfold the very tightly folded polypeptide, so it loses its shape. Shape is the most important thing when you're describing this side of the graph. If its shape changes, then its tertiary or quaternary structure will change. The active site will no longer be the correct shape, so it will no longer be complementary to the substrate. Now, that should be fairly easy for you to understand, should be easy for you to remember from GCSE. So we're talking at the peak of the graph there, I've just put a little thin line, that's the maximum rate that this enzyme has reached. But if we come down from that, we can mark that as the optimum temperature. That's the fastest temperature for that enzyme. That's not where the collisions are highest, because collisions just keep getting faster and faster and more the hotter you make it. But at that point, the collisions are very fast, but they're also successful because the shape is still correct. Now, what I want you to think about, linking this to the last video, consider how this graph fits with the lock and key versus induced fit theories of enzymes. If lock and key was correct, and that the active site had to be 100% complementary to the substrate, then the second any hydrogen bond started to break and it changed even slightly in its shape, it would no longer be complementary, and you'd cease all reactivity. Well, that's not what happens. Look at the graph. It curves down slowly on side B. Well, it's relatively quick, but you can see there's still quite a lot of distance there from the optimum temperature to zero activity. So induced fit looks more likely, that it will tolerate some level of change, some level of unfolding and denaturing, but once it hits a certain amount of denaturing, that's it. There's no more reactions that are going to happen. So this graph is good evidence for the induced fit theory, but you've got to be able to describe both sides of the graph using all this detail and all that detail, lots of key terms to get that right. We'll quickly whiz through an example of how I think you should do that in the exam, but take a screenshot of this if you're going to add it to your notes. Okay, let's move on. So if I was going to put that into words in the exam, for how does temperature affect enzyme activity, you're not likely to get it that specific. You'll get a graph and it'll say, explain what's going on here or what's happening at point X or whatever. But all you've really got to talk about is that at the start of the graph, with increased heat, the enzymes and substrates have more energy, so move more, have more collisions. Substrate collides with an active site that can form an enzyme substrate complex and turn the substrate into products. As the heat increases, so do collisions, so the enzyme activity also increases. All of the words I've put in gold, those are keywords, those will be in the examiner's mark scheme. So the rate of reaction will keep getting faster and faster until it reaches the optimum temperature. That's where the collisions are happening very rapidly, but before the active site loses its shape. That then takes you to the downwards part of this graph, that as heat continues to increase, it breaks the hydrogen bonds that hold the tertiary/quaternary shape of the enzyme together. The enzyme is denatured, which means the polypeptide chain of amino acids has begun to unfold, which changes the shape of the active site. When changed sufficiently, the induced fit cannot be achieved. The active site will no longer be complementary to the substrate. So even as collisions continue to increase, no enzyme substrate complexes can form, so no product is made, and the rate of reaction drops rapidly. Okay. Into full answers like this. The temperature tends to be the one that comes up the most. So we've spent a bit of time on that. We're going to whiz through these next ones. So pH is relatively straightforward. Okay, so you should remember from chemistry the pH scale that looks like this, with 0 over there on the far side. 0 is for strong acids, 14 close to me for strong bases or alkalis. Those are the colours that universal indicator will change. Now, what you may not know, or you may have forgotten from GCSE, is what makes an acid and what makes an alkali an alkali. And it's that acids give off high amounts or lots of hydrogen ions, H+ ions. So think about the acids you know, hydrochloric acid, HCl. In solution, it breaks apart into hydrogen and the chlorine, so it gives off hydrogens. So half of it are hydrogens that it's going to give off. Sulfuric acid, H2SO4. H2, two hydrogens, they're going to break off. You're giving out acids. Carbonic, H2CO3, you're giving off the hydrogens. All acids give off H+ ions. Those are the acidic part that does the damage. With alkalis, it's OH ions that they give off, hydroxide ions they give off. So think sodium hydroxide, lithium hydroxide, potassium hydroxides, all these alkalis that you've probably used in the lab before. They are really alkaline because they give off these OH- ions. Now what I need you to remember for enzymes is basically just acids and alkalis give off these charged particles. Now, why that's relevant is when you think about protein folding. If I've got a long polypeptide chain over there, but let's just kind of substitute that for this long blue line. That long blue line is representing my long polypeptide. That's got to fold up in a very, very specific way. I'm summarizing here. We've gone through primary, secondary, tertiary structures. To get the enzyme, we'll just put the outline of the enzyme there. There's the enzyme, there's the active site, there's the substrate that's going to fit into that. All that should be really obvious to you as to how a protein folds. Now remember, I whizzed up the screenshot on the first slide about the protein folding. Go back to protein biochemistry if you're not sure about it. Those bonds to give you that structure are dependent on the amino acids linking up when they fold. So either the little double bonded oxygen from the carboxylic acid group or the single bonded hydrogen from the amine group of adjacent amino acid, they've got positive and negative charges. So when it folds up, they can form hydrogen bonds to give you secondary structures. In the tertiary and quaternary structures, it's the R groups interacting, but a lot of those interactions are ionic bonds. So again, we're talking about ions, we're talking about charges, we're talking about attractions. If you throw a load of hydrogen ions, positives or negatives with the hydroxide ions in, those are going to mess about with where those folds and attractions can take place. So whilst an enzyme or any protein will form its correct folding at its correct pH, if you put it in its wrong pH, the H+ and OH- ions in the acids and alkalis, this is from over there, interfere with the bonding that occurs when a protein folds. So what you'll get in the wrong pH is it might fold up into this, which if I put the outsides on, you can see it doesn't really have outsides. Remember, we just add that to make the diagram more obvious. You can see that this one is very different to that one over there, even though it's from the same polypeptide, but it folded into a different shape because the positive H+ and OH- messed about with the attractions that could form in the folding. In other words, enzymes will only form their correctly shaped active sites when they're in their optimum pH. There is also an optimum pH as well as an optimum temperature. Hopefully that's quite straightforward. Take a screenshot of this. We can go really quickly with concentration because it's really simple and it's basically about collision theory again. But if I've got situation at A here within this box, and if within this box I've got one green guy and I've got one blue guy, let's say one of them's an enzyme, one of them's a substrate. If they're just bouncing around in that box, just passively in any direction, no kind of sense of what to do, then the chance of them bumping into each other is quite small. Because there's not many of them in there, it's a low concentration. If I put a situation B up here, where I dramatically increase the concentration of one of these things, let's say we're increasing the concentration of the blue one, let's say the blue guy is representing substrate molecules, okay? Then there's a much, much higher chance now that the green guy, who would be my enzyme, is going to collide with one of those blue guys once they start moving about. Simply by having more there, you've increased the chance of two bumping into each other. If they bump into each other successfully, form an enzyme substrate complex, you're going to get a reaction. So increasing the concentration obviously will increase collisions and increase the rate of reaction. Now, just to give a situation C on here, I'm going to take the same two guys from the first one, but I'm now going to put them in half the amount of space. What you also need to remember is that concentration can be affected by the number of particles or the space that they have to move around in. By halving the amount of space here, I've effectively doubled the concentration in C relative to A. Because there's less space, they're twice as likely to bump into each other. So it's not just about the number of particles, it's the amount of space they have to move around in, which of course is what concentration is a measure of. Just as an extra point, with the reducing the volume, of course, you can squash the volume of something, and we have another word for that, which is pressure. If you squash things down, it's pressure. But increasing pressure and increasing concentration are basically the same stuff. It's packing more stuff in a smaller amount of space. Increasing pressure is basically the same as increasing concentration. Okay, they're more likely to bump into each other. More collisions means more reactions. So any enzyme activity is fastest at high concentrations and high pressures. It's why in lots of industrial processes they do it under pressure, because it makes it go faster. Last one, let's talk about inhibitors now. So there's two types of inhibitors. There's competitive inhibitors and there's non-competitive inhibitors. The word to inhibit means to stop something from happening. So let's focus first just on competitive inhibitors. Now, if I bring up my enzyme and my substrate here, which we've seen so many times throughout my slides, the substrate fits in the enzyme because the active site is complementary. You know that. Let's just pop it back out again. If there's going to be a reaction, you need that to happen. But what if I've got these guys? So these are competitive inhibitors. Now, they may not look like much looking at them on there, but if you look closely at the competitive inhibitors there, you might notice something. If I remove one of them and pop it into location, look, those competitive inhibitors are complementary to a part of the active site. You can see I've just slotted it in there. So that competitive inhibitor has now effectively blocked the active site, meaning if the substrate was going to try and get in there, it wouldn't be able to fit. So this is why they're called competitive inhibitors, because they compete with the substrate for the active site. Now, if the enzyme's got a competitive inhibitor in its active site, it's clearly not going to form an enzyme substrate complex, so you've reduced the reaction. Now, this isn't a permanent binding. Okay, it's as if you've got the enzyme there, you've got the substrate or the competitive inhibitor, they both want it. If the competitive one inhibitor gets in there, it slows the reaction. If it then comes back out, maybe the substrate could go in there, but because it couldn't go in there straight away, it's had to slow down, or maybe a different competitive inhibitor goes in there. If there's loads of competitive inhibitors and not many substrates, then the chance of a substrate finding an empty active site is really low. So the more competitive inhibitors you've got, the lower the reaction will be. Okay, so the competitive inhibitor will bind to and block the active site, preventing the substrate from binding to form an enzyme substrate complex, and thus prevent products from being formed. Now, as I've tried to explain there about the numbers though, competitive inhibitors can be overcome by having an excess of substrate. I mean, excess means high amounts, way more than you need, because that reduces the chance of the inhibitor binding. If you've got one enzyme and 10 substrates and one competitive inhibitor, well, it's 10 times more likely that you'll get an enzyme substrate complex than an enzyme competitive inhibitor complex. So if you put loads of substrate in there, then you could make a situation where effectively it doesn't matter that the competitive inhibitor is in there. It's all about probability. The more competitive inhibitor there is, the slower the reaction is going to be. Now let's compare that with non-competitive inhibitors. Okay, so still same enzyme, same substrate here. But what we need to talk about now is something that you probably, well, certainly doesn't come up at GCSE. And it's on an enzyme, the active site is kind of the business part. That's where it needs to do its job. But there's loads of dents and bumps all around an enzyme where other things can bind. So if I introduce one of those over here, massively exaggerated on mine, but that's called an allosteric site, a kind of secondary binding site for other random things. I've relabeled active site on there just to compare the active site and the allosteric site. Now this is important for non-competitive inhibitors, because if I put some non-competitive inhibitors over there, you should be able to see fairly obviously that those non-competitive inhibitors I've created to specifically fit into that allosteric site. So if one of them binds into the allosteric site, what happens there is it kind of binds in. If here's my enzyme, okay, if a non-competitive inhibitor binds here, it binds in and kind of squashes the enzyme. It bends the shape of it. So if you look at the active site on this enzyme down the bottom now, it's been squashed and crushed. The substrate is no way going to fit in there. Not even induced fit theory is going to help here. So the binding of the non-competitive inhibitor permanently, this is irreversible binding, bends the enzyme out of shape and changes the shape of its active site. So that effectively wipes out that enzyme from being able to function. Okay, non-competitive because it's not competing with the substrate for the active site. It's binding somewhere totally different. So a non-competitive inhibitor will bind to an allosteric site, not the active site. This causes a conformational change in the shape of an enzyme. Conformational change just means how it's put together. The change in active site means it's no longer complementary to the substrate, and this is irreversible, so the enzyme is now functionally useless, or in other words, it's inhibited. Okay, so competitive inhibitors do inhibit the rate of reaction by slowing it down because they're competing, whereas non-competitive inhibitors basically neutralize and wipe out that enzyme for good. So your body has to make more enzymes. So lots of poisons and toxins are non-competitive inhibitors, and they do so much damage in your body because they can bind to proteins, change the protein's shape, and effectively wipe out its function. Right, let's do a quick summary of this then. Let's just get at the main points of the following, of the things that can influence the rate of reaction on enzymatic process, be it catabolic or anabolic. Just put these up as a reminder from the previous video. But the four things that we've just talked about then are temperature, pH, concentration or pressure, and inhibitors. And one sentence for each just to help you remember them. So temperature, it's initially due to collision theory because you're giving it more enzymes, more energy, more movement, more collisions. But it's due to denaturing after the optimum temperature, and that's all about how the protein folding is affected. pH is due to the H+ and OH- groups interfering with the folding and bonding in the secondary, tertiary and quaternary structures, meaning the shape will be wrong. Concentration and pressure is all about collision theory, and inhibitors, we've just said, it's competitive or non-competitive. Either way, it prevents binding the substrate to the active site, either by blocking it, because that's competitive, or by changing the active site altogether. So it's your very basic summary, but you need to be able to explain all 4 of those with masses of terminology from protein biochemistry, protein structure, amino acid biochemistry, how it folds, how it bonds, the enzyme function and mechanism. There's so much stuff. That's the key thing at A-level to getting good marks, is not just writing answers that are correct, but writing answers that contain lots and lots of detail. Okay, from here, this kind of completes this initial fundamental biology section of the course. I want you to go to saccharide biochemistry. We look at glucose and glycosidic bonding. In previous videos, we've looked at lipid biochemistry when we did membranes. We've looked at nucleotide and DNA biochemistry when we did everything about DNA. We've just done protein biochemistry in lots of detail. Saccharide biochemistry is the last one that finishes up everything on cells and biochemistry. That's a massive part of the course completed. So go to this one, and that's pretty much all your fundamentals done, and a long way to getting a good grade on your A-levels. As I always say at the start and I say at the end, please do like this video if you found it useful. Do subscribe to my YouTube channel if you haven't done so already, and have a look at all the information that I put on Facebook and Instagram. Thank you.